Hi everyone, this is Miss Longley, and today we're going to be covering chromosomes. We will be covering objectives 2 and 3 today, so let's go ahead and get started. So the first thing that you'll need for this screencast is you will need your packet out. We are going to be covering page 4 today and learning some new vocabulary terms. Although these vocabulary terms seem very similar, they are different. And it may also be helpful while filling out this term table to also include your notebook, because we're going to be talking about items that may not necessarily fit right on this page. So let's go ahead and get started. So quick little review, what is a gene? So a gene is a section of DNA that contains the recipe for making one protein. And if you remember from a previous unit, we learned that DNA molecules contain not just one, not just 10, but hundreds, thousands of genes in a single DNA molecule. So keep that in mind. A gene is just a small portion of it. It's just coding for one protein. We know that bodies need to make hundreds and thousands of different proteins. Therefore, we're going to find hundreds and thousands of genes. So we have talked about how DNA replicates itself to prepare the cell in order to divide. So we're going to take a look at what that looks like before and after DNA replication. So the picture here on the left, you can see we have a nucleus and we have these 5 little strings, different colors. We got some green and some purples here. This is what DNA looks like before DNA replication. We have 5 molecules of DNA here. After DNA replication, remember, we are making a copy of every single DNA molecule. So as a result, we double the amount. So if we start off with 5 molecules of DNA, we are now going to have 10 total. Each molecule of DNA now has an identical copy. So what does that look like in real life? Well, it's kind of messy. So DNA, when it's in this stringy kind of format, it's called chromatin. I kind of like to think of it as a bowl of spaghetti. So if the bowl was the nucleus, the spaghetti noodles are the DNA molecules. The more DNA you have, the more spaghetti you have. And what gets really complicated with cell division is that it's really hard to split apart this DNA evenly and to make sure that identical copies are separated so that each cell gets its own set. So if you can imagine trying to take this plate of spaghetti and split it into 2 equal portions, it would be really complicated and you're likely to make mistakes. So the problem with this is DNA in the format of chromatin. It's messy. It's really hard for the cell to divide and make sure that it gets a copy of each. So let's talk about how do we fix this. So DNA, again, like this spaghetti twisted mess, that's called chromatin. What the cell is going to do is it's going to take every strand or molecule of DNA, chromatin, condense it and twist itself into individual structures called chromosomes, which looks like this. You may have seen these before. They kind of look like X shapes. So chromosomes, a lot easier to maneuver and manipulate when the cell is actually dividing. So you can see in this plate here with that spaghetti noodles all twisted together, the 2 identical copies are intertwined and made into a chromosome. All right, so let's take a look at what a chromosome actually is. So a chromosome is DNA that is tightly coiled up around proteins called histones, kind of like when you take a string and you wind up a yo-yo. The yo-yo is the histone, the string is the DNA. So it's organized, it takes up less space. Again, it's easier for the cell to divide and split those evenly. Keep in mind that a chromosome carries genetic information in the format of genes, which is important because in order for your newly made cell to do its job, it has to have the instructions for making proteins. So in this unit, we are going to be focusing on chromosomes. Keep in mind that a chromosome contains a DNA molecule that's just been twisted up and organized. So With chromosomes, there are two different types, and based on the shape, it'll indicate to you whether a cell is preparing to divide or if it just finished dividing. So I would recommend drawing this in your notebook so that you have these two pictures available to you. So this picture right here with the chromosome as an X shape, this is called a single chromosome, known as a replicated chromosome, and that just lets us know that it contains two identical copies of the DNA as a result of DNA replication. So DNA replication has already happened. We have the DNA molecules all twisted and organized together in this shape, which is the X-shaped chromosome, otherwise known as replicated chromosome. So DNA looks like this when a cell is preparing to divide. It hasn't divided yet, it's getting ready to. After cell division, this is what a chromosome looks like afterwards. Again, it's still a single chromosome, but we call it an unreplicated chromosome. Why? Because now it's only containing one DNA molecule instead of two identical ones. They've been split apart because the cell was dividing. So when you see DNA that looks like this, that tells you that the cell has just finished its process of dividing into two new cells. All right, let's take a little bit more look at chromosome anatomy. So right here, this red structure that you see, this is a replicated chromosome. How can I tell? Well, it's in that X shape. B is pointing to a new structure. This is called a centromere. It's kind of like a belt loop that holds together this replicated chromosome, and it attaches the two chromatids together. You might be wondering, what is a chromatid? That's our next definition we're going to cover. So a chromatid is basically one side of this replicated chromosome. Keep in mind, replicated chromosomes contain two identical copies of the same DNA molecule. And so therefore, two chromatids are identical here as a result of replication. They're identical, which is where they get the name sister from. So these are two sister chromatids connected together that form one single replicated chromosome. So keep in mind, sometimes kids get confused as to whether this is two chromosomes or one. It's two sister chromatids, but it's still one single replicated chromosome. All right, let's take a look at types of chromosomes and how they exist in different cells. So I would recommend making a table in your notebook. We're going to be comparing prokaryotic cells versus eukaryotic cells. Let's start with the prokaryotic cells first. So prokaryotic cells are bacteria. They contain their DNA where it's floating inside of the cytoplasm. They do not have a nucleus, so their DNA is just floating around. The other thing about prokaryotic cells is they are very simple cells. They only contain one chromosome, and that chromosome is in a circular loop, so it's actually connected together, and it's just floating around in the cytoplasm. So they only have one chromosome. Eukaryotic cells are a little bit different. We're a little bit more complex. So our DNA is protected inside the nucleus. And again, this applies to all eukaryotic organisms. So a eukaryotic cell has many chromosomes, not just one, but many of them, and they're linear. And what that means is that if you were to unwind a chromosome and line it up straight, there would be a beginning and an end. That is what linear is referring to. And like I said, every species has a different amount of DNA. If you compare a human to this flower, you'll notice that the flower has way more chromosomes than we do. So here's just a short little table that will show you the different numbers of chromosomes found in various eukaryotic organisms. You can see a fruit fly only has 8, a tomato has 24, a human has 46, and really close to us is the chimpanzee. They have 48. So what you may notice is that eukaryotic organisms usually have an even number of chromosomes in their body cells. So body cells referring to liver, stomach cells, skin cells. Different species have different numbers. We'll cover this in our next unit, but just know that having an even number of chromosomes is very important in terms of cell division. So let's take a look at the human chromosomes. So again, we have 46. Where did those 46 come from? Well, you may know that half came from mom and half came from dad. So 23 of your chromosomes came from mom and 23 of the other chromosomes came from dad. When you add those up together, that's where the 46 comes from. All right, and our last slide for this lesson. Homologous pair of chromosomes. This is a tough word to say, so you may need to practice it out loud. So it's called homologous chromosomes. So homologous chromosomes are chromosomes that are the same size and they contain the same genes. So for example, they contain the same gene for hair color. So you get a gene from mom for hair color, and you get a gene from dad that contains maybe a different hair color. So in your body, you have two different sets of genes. This is considered a homologous pair. So chromosomes that contain the same gene is part of the pair. One comes from one parent, the other comes from the other. You'll notice here in the picture, the same size part. Notice how I paired the purple chromosome from mom with the pink chromosome from dad. They are the same size. They also contain the same genes, such as hair color. Therefore, these two collectively are called a homologous pair. We can see that these blue, so the darker blue and the lighter blue one comes from mom, one comes from dad. They're the same size. Therefore, they're probably containing the same genes. That is a homologous pair. What would not be a homologous pair would be this purple one with this green one here. One, they're different sizes, and two, they're coming from the same parent. So it has to come from different parents, and they have to have the same genes and the same size. All right, that is today's lesson. Thank you for watching. Make sure that you have all the terms and vocabulary filled out in your packet and that you have any of the other notes written down. Thank you for watching.